Hello and welcome to another episode of Literature Talk. Today we have with us Annam Surendra Kumar. He completed his MSc from Andhra University and is currently pursuing his PhD in organic chemistry at IISc Bangalore. In today's presentation, he'll be discussing an exciting and important area of research, nanometal surface energy transfer for biosensing and bioimaging. So Surendra, welcome to the talk and the screen is yours.
Okay, thank you, Baya. Thank you. And welcome to the turning point. So before I begin, I would like to sincerely thank you, Baya, for creating this platform for literature talks and giving me this opportunity. So today I will discuss about this nanometal surface energy transfer for biosensing and bioimaging applications. So mainly this technique provides an alternative energy transfer pathway to overcome the distance related issues we are facing in resonance energy transfer. So with that overview, let me briefly outline the structure of my presentation. So in this presentation, I will briefly discuss the importance of biosensing and bioimaging. Then I will discuss the resonance energy transfer, and I will introduce this nanosurface energy transfer, and I will present the experimental investigations that validates this mechanism. And finally, I will highlight the applications and future perspectives of nanosurface energy transfer and concluding with a brief summary. So first, let me begin with the importance of this biosensing and bioimaging. So as we know that in vivo and ex vivo fluorescence imaging of live cells using these fluorescent materials has become an essential tool in modern biomedical research. So it plays a crucial role in understanding the cellular mechanisms in pathology and also in medical diagnostics. So because of these advantages, fluorescent materials having high sensitivity and high specificity and good photostability while causing minimum damage to the cell are attracting huge interest. So therefore, a wide range of fluorophores have developed and studied. So however, they still requires a special attention. So to address these challenges, advanced energy transfer mechanisms have been explored. So in this context, the nanometal surface energy transfer has emerged as a promising technique for long-range optical distance measurements. So before going in detail about this nanosurface energy transfer, it is conceptually equal to the resonance energy transfer. So it is better if you understand what is resonance energy transfer. So resonance energy transfer, also known as this Foster resonance energy transfer FRET. So it is a non-radiative process in which we can see that this is the donor molecule. This excited donor molecule will transfer its energy to the acceptor molecule through dipole-dipole coupling without emitting a photon. So for this process to occur efficiently, this donor and this acceptor molecule must be in very close proximity, typically in the range of 1 to 10 nanometers. Now here, if you look at the first image, this represents the donor emission. Here, first the only donor molecule is excited and we are observing its characteristic emission, which is shown in the purple region. And in the second image, we see the acceptor emission. This is corresponds to the fluorescence of the acceptor molecule. Now, in the third image, when this both donor and acceptor molecule here are present, and when they are in close proximity, when they're close proximity, you can observe as a result, this donor emission decreases completely, and we are observing the acceptor emissions predominantly. So this shift from this donor molecule to the acceptor molecule is a clear indication that is a resonance energy is happening from this donor molecule to the acceptor molecule. So to better understand this phenomena, let's look at the history of this resonance energy transfer. So the earliest concept of this resonance energy transfer was proposed by Professor Perrin, and he suggested that the energy transfers non-radiatively from a donor to the acceptor molecule. And later, Professor Foster developed a quantitative theory to explain this phenomenon, and he demonstrated that the efficiency of energy transfer depends very strongly from the distance between this donor and the acceptor molecule. So because of this fundamental contribution, he is well known as the father of resonance energy theory. Often we call it as a FRET. So now let us understand how this resonance energy transfer mechanism actually works. So we can see here initially the donor molecule is in its ground state. So when it's absorbed the light energy, it's get excited from the ground state S0 to the excited state S1. So after excitation, the donor undergoes a vibrational relaxations and comes to the lowest energy level of the excited state. And in the absence of acceptor molecule, the donor will flaxes back to the ground state by emptying a fluorescence. So which is known as, we can say normally, it is a donor emission. But when acceptor molecule is there in the range of 1 to 10 nanometers, and we can see the left right side, this donor emission spectra is overlaps with the acceptor absorption spectra. So instead of emitting light, it transfers its energy to the acceptor molecule through dipole-dipole coupling. So this process is known as resonance energy transfer. And as a result, we can see the acceptor gets excited to the excited states and relaxes back to the ground state by emitting a fluorescence via radiative or a non-radiative pathway. So for this process to occur efficiently, there must be a significant overlap between this donor emission spectra and the acceptor absorption spectra. And according to this Foster theory, here we can see the rate of energy transfer is given as follows, where the R is the distance between this donor and acceptor molecule, which follows a 1 over R power 6 distance dependency. So because of this strong distance dependency, this resonance energy transfer has a wide range of applications in biology. As we can see here, the resonance energy transfer is used in this biosensing for detecting biomolecules and in extensive use in live cells for imaging or to track the molecular events in the real time, and also used in these protein-protein interactions. Here you can see when these two proteins interact by a donor and an acceptor molecule, when they came to the close proximity, we can see here the resonance energy transfer. As a consequences, we can observe the binding between these proteins. And as well as it is widely used in to study the conformational changes of proteins and biomolecules. Instead of having these many applications, this resonance energy transfer has several limitations. As we can see here, the efficiency of energy transfer decreases rapidly with increasing the distance. As shown in this one, we can see it is follows an inverse power 6 distance dependency. So this means even a small increase in the distance, which leads to the drastical energy reduction and energy transfer efficiency. And second one is in resonance energy transfer, it is a highly sensitive to the orientation of this donor and acceptor dipoles. For the efficient energy transfer, these two dipoles, the donor dipole and this acceptor dipole, should be aligned in a parallel way or a favorable way. And if they are aligned in a perpendicular, you cannot observe the resonance energy transfer from this donor to the acceptor molecule. So because of these limitations, this resonance energy transfer becomes ineffective for the larger biomolecular assemblies, where the distance often exceeds beyond the 10 nanometers. So there is a need of an alternative energy transfer mechanism. As we can see here, also to overcome these distance limitations and these orientation factors. So to overcome this, researchers developed a new technique. They developed this nanosurface energy transfer, where the need for long range sensing is, and one more is by avoiding this orientation constraints, they developed this nanosurface energy transfer. So this mechanism, we will go to the in detail how this nanosurface energy transfer will works. As we discussed previous, it's conceptually related to the resonance energy transfer. Here, initially the donor molecule is there, so it absorbs light energy and gets to the excited state. So instead of transferring its energy to the another molecule, here the acceptor has a metal nanoparticles, typically the gold nanoparticles we are using, and the excited donor transfers its energy to the acceptor molecule non-radiatively to the surface of the metal nanoparticles. And this process occurs through dipole, here the donor dipole and this metal surface. So because of this mechanism, it will be effective till up to the 40 nanometers. And according to this nanosurface energy transfer, we can see the rate of energy transfer as follows, where d is the distance between this donor dipole and the acceptor molecule. So previously we discussed this resonance energy transfer where the distance dependency is 1 over r power 6. So here we can see the distance dependency is 1 over t to the power of 4. So now the question naturally rises, how this energy happens from this donor dipole to the acceptor particles. So when we consider a bulk metallic particles surface like, so this energy transfer is governed by the phenomena known as the surface plasmon resonance. So this surface plasmon resonance is a collective oscillations of free electrons at a metallic dielectric interface. So this collective oscillation of electrons generates a electromagnetic field around the surface of the metal, and it will interact with the donor dipole, and as a consequences we can see the energy transfer from this dipole to the metal nanoparticles. But when we move to nanoparticles, this phenomena is known as localized surface plasmon resonance. So in metal nanoparticles, such as we can see the gold nanoparticles. So what happens here? Here the free electrons are confined within a small volume due to its nanoparticle behavior. But when the light interacts here, the electron cloud oscillates collectively around this nanoparticle. So because of this, the electrons are confined and they will generate a highly strongly localized electromagnetic fields. So this highly localized electromagnetic field will interact with the donor molecule, also and which strengthen the interaction between this excited donor molecule and the metal nanoparticle. So, and this is the fundamental reason why this nanosurface energy transfer can efficiently work over the longer distances. So as we have seen the previous slides, we can clearly say that nanosurface energy transfer is somehow better than this resonance energy transfer. So here the resonance energy transfer is happening through between this dipole-dipole interaction, where in case of this nanosurface energy transfer, it is a dipole to a metal surface interactions. And as a consequence of this distance dependency of 1 over R46, the sensing is limited to the 10 nanometers. But we can see here this nanosurface energy transfer will sense till the 40 nanometers. And in contrast, here we can see the resonance energy transfer requires a strong overlap between this donor and acceptor emission, and the orientation sensitivity should be considered. But in case of this nanosurface energy transfers, these are exempted. This is not that much dependent upon the spectral overlappment and this orientation sensitivity. And another important limitation here, you can see the nanoparticle size. So there is a no need of this nanoparticle size in case of resonance energy transfer. But this nanosurface energy transfer will highly dependent upon this nanoparticle size. If the nanoparticle size varies, the energy transfer efficiencies and the distance of this energy transfer efficiencies varies. And coming up to the investigations of this nanosurface energy transfer, I will discuss how energy transfers non-radiatively from this donor molecule to the metal nanoparticle, and I will also discuss the distance and size dependent sequencing, and how this nanosurface energy transfer fundamentally differ from the resonance energy transfer. And in the following slides, I will explain the experimental designs and the key results that confirms this nanosurface energy transfer mechanism. So to begin with, let us look at the landmark study that established this concept of nanometal surface energy transfer by Strauss and co-workers. So in this study, they used this fluorescein emirate or FAM as a fluorescein donor molecule. On the right side, you can see the gold nanoparticles are used as an acceptor molecule, and they systematically varied the distance between both this donor and the acceptor molecule. And to achieve this, they designed a well-controlled experiment system. You can see here, both the donor and acceptor are separated by a double-stranded DNA. So one end is functionalized with fluorescein, and the second end they functionalized with the AU nanoparticles, and varying the different DNA lengths from 15 to 20, 30, 60 base pairs, which are correspond to the distance of 6 to 23 nanometers. So, but before looking to the results, it is important for us to understand how this AU nanoparticle and a dye are linked to this double-stranded DNA. Here, the system is constructed using these two single-stranded DNAs. Okay, one is functionalized with the donor dye and another one is functionalized with the gold nanoparticle. Here we can see the first, the fluorescent dye. It is attached to the DNA using this succinimidal ester functional group, which is known as FAM ester. And similarly, we can see here the gold nanoparticle was attached to the double-stranded DNA with the modified DNA group. So after getting these two strands of DNA, this is one of the strand and this is strand B. And finally, on hybridization of these two strands, we get this double-stranded DNA duplex. So finally, this to confirm the energy transfer efficiency through the distance dependency, they studied on controlled experiments. Here you can see here the donor molecule like FAM and the acceptor molecule nanoparticles are separated by a double-stranded DNA. So to test the distance dependency, they introduced a specific DNA binding protein known as MECORE. So which recognize the selective site of this GATTC at this double-stranded DNA, and it makes it bend from 128 degrees. So this binding effectively reduces the spatial distance between this both donor molecule and the AU nanoparticle. So as a result, we can see the energy transfer efficiency increases from leading this fluorescence quenching of this donor molecule. So now let us see how the energy transfer efficiency varies with the distance. Here we can see the energy transfer efficiency of the plot of here the separation distance. On the you can see the red plot, the dashed line where it indicates the resonance energy transfer, and it represents the very sharp drop due to its 1 over r/6 distance dependency. And in contrast, we can see the solid curve, which represents nanosurface energy transfer, which follows 1 over d/4 distance dependency and decays more gradually. So this one clearly shows that nanosurface energy transfer operates over longer distances compared to the resonance energy transfer. And experimentally, they varied the DNA lengths by controlling distances as the distance is increasing from 15, 20, 30, and 60 base pairs. We can see the quenching efficiency. So importantly to note that even at larger distances, this nanosurface energy transfer shows a measurable energy transfer. So now let us understand the fundamental origin of this behavior using this UV visible spectroscopy and the theoretical modeling of this AU nanoparticle cluster. Here we can see the plot of absorption spectra between this, here the curve, the dashed line we can see here, and this represents a absorption spectra of calculated AU nanoparticle, dye, and the double stranded DNA. So this is the real experimental spectra. And this semi-empirical fit is the combination of two spectra. One is the ligand alone, and one more is the calculated A nanoparticle spectra. So after combining this semi-empirical fit and the experimental data, we can clearly see the overlap, excellent overlap between these two. So this excellent overlap, which indicates the A nanoparticle behavior for this optical behavior of A nanoparticle. To further validate, they supported the comparison plot where the resonance energy transfer shows a sharp peak at the shorter distances. So we can say here clearly the resonance energy transfer is highly sensitive compared to the surface energy transfer, which shows a broader peak and extended distances responses. And to further validate these mechanisms, Strauss and co-workers extended their study using different fluorescent dyes and then monitored the fluorescent lifetime and photoluminescence quenching. Here you can see they used this red fluorescent and one more FAM dye by carefully controlling the distances. And here we can see the schematic representation of these gold nanoparticles and the dye molecule attached to the double-stranded DNA. On the right side, a simplified model shows a donor interacting with a nanometal acceptor over a distance of separation of d. And as previously discussed, in case of resonance energy transfer, the energy transfer happens from this donor to the acceptor molecule through a dipole-dipole interaction. But in case of this nanosurface energy transfer, the energy transfer happens from this metal nanoparticle from donor to the metal nanoparticle through dipole metal surface interactions. So these A nanoparticles have these free electrons around the surface, which generates extended electric fields. Here we can see the gold nanoparticles in an idealized electric field. So this electric field enables a long-range coupling between this donor and the metal surface. So to further validate these studies, they did the fluorescent lifetimes. As we can see the plot of this fluorescent and AU nanoparticles. Here as the donor, this is this donor molecule and this is the acceptor molecule. And this is the distance between this. We can see as we decreasing the distance from 45 to 15 mer, the fluorescence of lifetime is decreases. So which indicates the non-radiative energy transfer from the dye to the nanoparticle. Similarly, the same trend they observed for the another dye where the distance is decreasing from this 45 to 15 mer, they observed the decrease in the lifetime. In addition to this fluorescent lifetime, they did the photoluminescence quenching, which is also shows a similar trend for both this FAM and the cyanide dye. We can see the decrease in the intensity as we decreasing the distance from the 45 mark to the 15 mark. And the photophysical properties further supports these studies where the photoluminescence quenchings and this lifetime of the quenching efficiencies relate 15 base pairs. As we decreasing the distance from 45 to 15 mer, we can see the decrease in the photoluminescence quenching and the lifetime of the quenching efficiencies. And finally, to prove this, so this is the finally, it is following a non-radiative pathway, not a radiative pathway. So they did the absorption spectroscopy to prove this mechanism is entirely a non-radiative pathway. So furtherly, you can see normalized absorption spectra. There is a small change in the observed spectra. So which suggests a minor change is due to this radiative pathway. But this majority of these curves didn't change. So which indicates itself it is a non-radiative pathway. If it is a radiative pathway, we can see the change in the absorption spectra due to the change in the oscillator strengths. And the further they extended these studies, they investigated the size dependency of nanosurface energy transfer behavior. But the same group came up with here we can see the plot of quenching efficiency as a distance. As quenching efficiency is decreasing if we decreasing the distance, if we increasing the distances. So this means the nanoparticles from 1 to 8 nanometers, the longer nanoparticle shows a transfer of energy more efficiently over longer distances. So this larger nanoparticles have a stronger electromagnetic fields. So which is why it is showing a quenching at a longer distances and a high quenching efficiencies. So due to these unique properties of this nanosurface energy transfer, it has a wide range of applications in medical diagnostics. Here you can see the nanosurface energy transfer. In case of protein interaction, we can study these proteins through this fluorescence quenching and recovery. And in biomolecules, if we see, we can monitor the small biomolecules and enzymatic reaction. And in case of nucleic acid, it allows us ultra-sensitive detection of this DNA and RNA sequences through hybridization-induced fluorescence quenching. And also in nanomedicine, it is used to track drug delivery of nanoparticle distribution and therapeutic activity with the biological system. And due to the time, I will discuss one of these applications which is reported by the Lin and co-workers. They developed a nanoparticle probe. So in this design, they used two DNA strands. One is here, we can see Tamara labeled DNA, and one more is this thiol blocker DNA. So initially, these two are the single stranded. So after this one, after hybridization, they got this double stranded DNA. We can see the fluorescent turn on, and they added A nanoparticles. As a consequences, we can see the nanosurface energy transfer from this dye to the nanoparticle. So now it is in a completely turn off state. So they did the studies. So before apoptosis process, the microRNA is not present. So the reporter remains bound to this nanoparticle and stays close. It's a signal off state. And during the apoptosis process, we can see here the microRNA concentration is increasing. As a part, this microRNA will selectively bind to the reporter molecule and this blocker will move far away. So as a consequence, the nanoparticle and this dye moves far away from this distance, and we can see the fluorescence turn on. So in this process, they did imaging. To study here, they did the absorption spectra of this A nanoparticle with the linkage of DNA. So they confirmed this A nanoparticle linkage with the DNA using this red shift in the absorption spectra from 518 to the 522 nanometers. And furtherly, they did the fluorescence luminescence intensity, where this is the right side, we can see this is the Tamara labeled DNA. Initially, it has a fluorescence lifetime of maximum. So when it is attached to this A nanoparticle, you can observe the complete quenching of this dye due to nanosurface energy transfer technique. And one more here, during the process, before we observed in the during apoptosis process, the microRNA concentration is increasing. So as the consequences, we can see this A nanoparticle is moving away from the dye, and we can see the recovery of this fluorescence dye. To further validate, they did the fluorescent lifetimes. It is clearly say the evidence where during the Tamara DNA initially has a maximum lifetime. So it is a it's free state. But when it is attached to this A nanoparticle state, we can see the decrease in the lifetime. So which indicates the energy transfer happens from this dye to the nanoparticle. And one more, when it is during the reaction process, we can see the microRNA detection is there, and microRNA is replacing this donor molecule. As a consequence, the fluorescence will be turned on. So we can observe the fluorescence as a recovery state. And they included in a real-time expression where they did microRNA 630 in the A549 cells. Here we can see clearly in 0 minutes. So they used this nanoprobe for the studies of imaging. So in 0 minutes, it is completely off state. So we can see there is no fluorescence turn on. So during the process from 30 to 120 minutes, the fluorescence recovery is increasing. So which is why we can say the microRNA concentration is also increasing. As a consequence, the dye move far away from the A nanoparticle, and we can observe the fluorescence recovery. And one more group is using this nanosurface energy transfer, they developed a hepatitis C virus sensing using this complementary RNA and DNAs. So initially, they used this DNA with a cynin 3 dye and a nanoparticle. So it is in now, initially it is in off state, but when it is find its complementary DNA, it will bind to the DNA, and as a consequence, distance is increasing and the fluorescence will be recovering. So these are the applications. As we can see the more applications. This nanosurface energy transfer has a wide range of future prospects. This nanosurface energy transfer we can include in a plasmonics and nanophotonic architectures for point-of-care diagnostic devices. And we also include this artificial intelligence in nanosurface energy transfer for to study these complex bunching profiles for improved data accuracy and can monitor the continuous. And one more future prospect is here we can develop implantable and wearable nanosurface energy transfer sensors for this personalized healthcare. So finally, to conclude the what I discussed, I discussed the introduction of resonance energy transfer and nanosurface energy transfer. Then I discussed the investigations on this nanosurface energy transfer, how the researchers validated this mechanism, and introduced the applications and the future prospects. Finally, thank you everyone.
Nice presentation, Surendra.
Yeah, thank you.
If you enjoyed this session, don't forget to like, share, and subscribe. We'll meet you in the next episode, next session. Thanks for watching.